Emmetropization, anatomy and biology. As we spoke about previously, emmetropization is the process by which the eye controls its growth to optimize vision. To do that, it must first encode the sign of defocus. As we previously spoke about in other lectures, the sign of defocus is where the image is placed relative to the retina. Myopic defocus, or if the image is placed in front of the retina, sends a signal for the eye to shorten or to cease growth. Conversely, hyperopic defocus, or placing the image behind the retina, is a signal for further elongation of the eye. And it does that through a number of biological chemicals within the system. Here's some starting retinal signaling molecules that possibly identify components to the emmetropization process. Those include dopamine, VIP, glucagon, GABA, nicotine, and melanopsin. As an example, nicotine itself is a very powerful inhibitor of growth in animal models. Nicotinic receptors exist in the retina during development, and they're also present in the ciliary body. And what we do see is actually the presence of nicotine through smoking affects the refractive development of children. Whether during the womb or during development, if children are around smoke, they tend to exhibit less myopia or relatively more hyperopia. Having said that, don't recommend smoking for your kids to stop myopia. Ultimately, these biological molecules come from the actual retina. They have to be detected by that retina and then sent out through the rest of the eye. Let's look at some layers of the retina and how they might actually be involved in the process. We're not really confident that ganglion cells right now play an important role in emmetropization. And that's largely because we know we can cut the optic nerve, destroy those cells, and the eye still responds to growth properly. The amacrine cells, though, and the bipolar cells, there are special classes of amacrine cells within the internuclear interplexiform layers that have been shown to be differentially sensitive to hyperopic and myopic blur. We also know that photoreceptors are a critical component to the densities for blur detection signals. And they're also a possible component to longitudinal chromatic aberrations, which is a theory of how the eye encodes sign. Whatever the cause in the signaling process within the retina, that signal must then go through the RPE, through further molecules, ultimately reaching the choroid. If the eye is going to elongate, that choroid needs to change in thickness. And so ultimately, this choroid must begin to thin as the eye lengthens. Here's an example on the left of 4 people with emmetropia in their choroids, and on the right, some high myopes in their choroids. If you look at these images, on the left, the emmetropic choroids are actually big and thick. That's these kind of hazy, splotchy areas that the blue arrows are pointing at the bottom. As the eye lengthens, though, the choroid ultimately starts to thin, so it's almost non-existent once we have high myopia of -10 diopters or more, as we can see in the right images. Ultimately, though, the signal then must carry on into the sclera for further remodeling. Just like in the choroid, as the eye lengthens, the sclera must also either thin as the eye lengthens or thicken if it's going shorter. If we get high enough scleral thinning, we can actually start to see it outside the eye as we have choroidal ruptures. Ultimately, though, this results in the eye growing in axial length, which is highly correlated with refractive development. Here we have examples of MRIs showing different eye lengths within the eye itself. At the top, we have a 7 diopter myope, and at the bottom, we have an emmetrope. And the eye size is the ultimate difference between the two that determines their refractive error. Finally, though, we have to go back to the retina, go back to the defocus, and check to see if this process should continue or we've completed emmetropization. This ultimately, though, has disease ramifications, especially in high myopia and high hyperopia. The stretching of the retina can lead to things like retinal detachment and macular holes. The thinning of the blood supply, including the choroid, can lead to macular neovascularization. And changes in the anterior chamber and the optic nerve can increase our risk for glaucoma. Thank you.